Hello, and thank you for joining me for today's Tech Talk. As you can tell from the title of my presentation, I'm going to be talking to you today about light sheet fluorescence microscopy and how we at Luxendo are working to create light sheet microscopes in order to help researchers like yourself with biological imaging experiments. To remind you who Luxendo is, we're a startup that spun out of the EMBL in September 2015, and we're a company dedicated solely to light sheet fluorescence microscopy and the development of the application in close collaboration with researchers like yourself. In 2017, we joined forces with Bruker, a world leader in high-performance scientific instrumentation. To set the stage for the rest of my talk, let's first consider some challenges in modern biological imaging. There's been an increase in the number of labs utilizing whole organisms, 3D cell cultures, and other physiologically relevant systems, and these provide a particularly unique challenge with regards to microscopy. First, researchers have to image extremely large volumes of three-dimensional data. Secondly, they need to capture this data at high enough resolution in order to localize the proteins and organelles of interest. Third, we need to capture this data quickly enough is to see the biology on physiologically relevant timescales. And last but not least, we need to capture this gently enough as to not perturb the system through light-induced photo damage, such as photobleaching and phototoxicity. And this is a particularly tall order for any sort of microscopy modality out there. Stereotypically, your microscope systems will fall short in one or multiple of these categories, and it's left researchers looking for more and looking for new and alternative ways to do their biological imaging. And recently, we've rediscovered a century-old technique called light sheet microscopy, and this has reemerged as a technique perfectly suited for these three-dimensional biological studies. What's unique about a light sheet system is the fact that we now decouple the fluorescence excitation pathway and the detection pathway into two separate, orthogonally, stereotypically, pathways. So what this means is that we'll have our sheet of light created via a laser beam here, which will selectively illuminate only the focal plane of interest of the detection lens in the detection pathway. This gives rise to the acronym SPIM, or Selective Plane Illumination Microscopy, also known as light sheet. But this still doesn't explain why this is such a great technique for biological imaging. To dive into this a little further, let's consider this cartoon Drosophila embryo here, and imagine I want to image this on a point scanner, a spinning disk, and a light sheet. If I'm interested in the focal plane highlighted in red, and I want to image that on a point scanner, I'll bring a laser beam in, a point source beam, which will travel through the sample, focusing to the focal plane of interest, and defocus on the backside. To create my image, I'll raster scan that laser beam, exciting one point at a time and collecting one point at a time on a PMT. In doing so, what I end up doing is exciting all the fluorophores in the sample with my excitation light, despite the fact that my image is only constructed of this small focal volume around the focal plane of interest that made it to my detector. The rest of this light above and below the focal plane was thrown away via a pinhole in a conjugate focal plane. So, fairly slow. We're doing a point-wise excitation detection. Spinning disk gets us a little bit of speed, and this is due to the fact that we parallelize our excitation. We have multiple laser beams exciting multiple points at a time, and now we parallelize our detection as well by using a camera instead of a PMT. However, the same drawback applies in the fact that we've now excited all of the fluorophores in our sample plane of interest, despite our image only being made up of this very small focal volume, the rest of this light being rejected by pinholes downstream. How light sheet differs is if I want to image the focal plane highlighted in red, I'll selectively illuminate only that focal plane highlighted in red, and illuminate all the fluorophores in that optical section in a single shot, and then can collect that entire optical section on a camera. This gives us a couple key advantages over confocal microscopy. First is speed. Because of the fact that we have the axial confinement of our illumination light, it means that we no longer need a pinhole in a light sheet system. What that means is that we have the detection efficiency of a wide field system with the 3D sectioning capabilities of a confocal, leading to being able to acquire images extremely quickly. The second benefit of this, and it might not be so obvious, is the lack of photobleaching and phototoxicity. We're using our fluorophores a lot more efficiently. You can see that in the schematic here, because we are not illuminating the fluorophores above and below the focal plane of interest, and because we don't need that pinhole, we're now exposing the embryos to significantly less amount of light, which leads to less photobleaching and less phototoxicity. To highlight this, here I'm showing you a Drosophila that's undergoing gastrulation. We've got the ventral side in the big inset and the dorsal side in the upper right corner. And we're imaging this Drosophila every 30 seconds for 15 hours, and then every 10 minutes for 5 hours, and essentially watching it undergo its development, something that you can't do on a point scanner or a spinning disk, at least not with this sort of temporal resolution. So the take-home messages for this and the big key advantages of light sheet compared to confocal is dramatically reduced photobleaching and phototoxicity. It's estimated that a light sheet system exposes your sample to about 3 orders of magnitude less light than on a laser scanner or a spinning disk. Additionally, we get much higher imaging speed with the detection of the optical section in one shot. And we can do this with the same or similar 3D optical resolution as we would get on a confocal system, making this ideal for imaging dynamic processes and live specimens. And just as there's a large diversity in biological questions and model organisms from the people that are listening to this talk, there's a large diversity in light sheet systems out there. In fact, light sheet can cover about 6 orders of magnitude from sample sizes, everything down to diffraction limited samples, all the way up to imaging an entire cleared mouse. However, just as there's not one microscope that always does the job, there's not one light sheet that does the job. And to highlight this, I'm going to utilize the Luxendo portfolio and just kind of give you an idea of where each one fits. So first up on the list is our Invi Lattice Pro. This is our Lattice light sheet. That's really geared for imaging things under 150 to 200 microns. We have our TrueLive 3D imager. This is a 3D multiplexed light sheet, really geared around organoids and spheroids. Our QuVI SPIM, which is kind of the jack of all trades. Cover slip mounted samples, everything from cells on a cover slip up to cleared samples. The Multi-View SPIM, our flagship light sheet. This was originally developed for work on Drosophila embryogenesis and zebrafish embryogenesis, so those are the key application areas for that. And last but definitely not least, our LCS SPIM, or large cleared sample, where it's just as it says, we're imaging very large cleared samples. So this big portfolio of Luxendo light sheets should help kind of show the diversity of these systems. Now, to get into these in a little bit more detail, for this particular crowd, I'd like to focus on our Lattice light sheet, our TrueLive imager, and our QuVI SPIM, because the application areas here, as highlighted on this slide, really fit well with kind of what the general theme of the conference is. These are not something I would say are set in stone. It's just more of guidelines, and I think the rest of the presentation we're going to go through each of these in general, and I'll give you a better feel for kind of what the strengths of each system are. First up on the list is our Invi Lattice Pro. This is an inverted light sheet, an inverted lattice light sheet at that. And the Lattice Pro is built up of a spatial light modulator, and this allows us to create a high number of different light sheet patterns out there and illuminate our sample based on kind of what we need out of our sample, whether that's a static Gaussian beam, a scanned Gaussian beam, a Bessel beam for a very large field of view, or a optical lattice for extremely high resolution. The Invi Lattice Pro is a two-lens geometry. It has a single illumination lens, the Special Optics 28.6, 0.7 NA water immersion objective. It's going to be key coming up. And our Nikon 25x 1.1 numerical aperture water immersion detection objective. Now 25x for cell biological applications isn't really kind of quote high mag, and so with this amount of NA we can mag up to 31 or 63x and still not be throwing away light. So we have a magnification changer in there that allows you to switch between these two magnifications. Two cameras downstream for dual color simultaneous imaging, and of course the spatial light modulator technology for creating our multiple different illumination beams. All this in a stable benchtop imaging platform that is fully computer controlled and what we would like to say is undergraduate friendly. Really not anything on here that you can screw up. To go over the system in a little more detail, we'll just go over the components individually. We have our incubation chamber as highlighted there, and the two scientific CMOS cameras, single illumination objective, single detection objective, and the two cameras for two-color simultaneous imaging. This is an inverted geometry, an inverted light sheet, and so the specimen carrier is a little bit different than what most people are used to. So I'd like to take a slide and go through that. So the sample mounting on the Lattice Pro is just slightly different than your standard microscope, and this is because we have water immersion objectives. So here, this is a side cutaway of this portion here, and we'll have our water immersion illumination objective and our water immersion detection objective in there. And as I've been hinting, we fill this entire chamber with water, which is how we get an inverted light sheet. However, if we're putting samples into this, most samples stereotypically do not like water. So how we get them in is we've developed a chamber with a biddy that has an FEP foil bottom. FEP is just the acronym for Teflon, and Teflon has a very similar refractive index as water. So we line the bottom of this with FEP. We can immerse that into this chamber, and now that creates a physical barrier between the water immersion, which we're using for our illumination objectives, and gives us a small volume of our immersion media for our specimen. Then we can either culture our specimen in these chambers or just physically drop it in, as in the case of this organoid here. And when we're ready, we shoot our light sheet out of the illumination objective, and orthogonally to that, we collect it on the detection objective. Alternatively, if your cells don't grow well on FEP or if there might be a potential problem, we do have a cover slip mount for this. Essentially, you get rid of the FEP, you get rid of the chamber, and now you fill this entire chamber with your immersion media instead of water, and then we can invert a cover slip in there. So let's get to some applications data. Here what we're showing is some epithelial cells that are seen for actin and chromatin, highlighting the high resolution of this system. However, one of the big advantages of this is for doing live cell imaging. So here we're showing you some HeLa cells that are undergoing mitosis with the tubulin stained in both green and the white, and allowing us to see, again, the extremely high resolution while doing live cell imaging. The next up is organoid that we imaged on the Invi. Here we are looking at tumorigenesis in a mouse mammary organoid. And what I'd like to point out about this is we were able to image this for 3 full days on the light sheet. And we're able to collect some pretty phenomenal data on something that it's hard to image for 3 days. So that was our Lattice Pro. Let's now move on to our TrueLive 3D imager. This again is an inverted light sheet, except now we've added a second illumination objective. We now have a 3-lens geometry here with 2 illumination objectives and 1 detection objective. These illumination objectives are a little bit different than on the Lattice Pro. We don't need the high numerical aperture because we're not creating any lattice light sheet patterns. And so we drop the NA, drop the mag a little bit, and are working with some 10x 0.3 numerical aperture water immersion objectives. Same detection objective as in the Lattice, magnified to 31 or 63x. Again, 2 cameras for 2-color simultaneous imaging, and acquiring these at greater than 80 frames a second for the full chip. The big difference in the TrueLive is the really large sample chamber, 75 millimeters. Gives you a lot of real estate to cover tens to hundreds of samples. And additionally, we can stick 3 of those TrueLive dishes in there that we showed on the Lattice light sheet slides. And again, just as before, up to 6 lasers, full environmental control, and a benchtop fully contained imaging system. So a side view of the TrueLive, we've got our 2 illumination lenses oriented on opposite sides of the sample. We've got our sample chamber here and our little organoid in the middle with our detection lens perpendicular from below. Again, that 25x 1.1 numerical aperture, post-magnified to 31 and 63x. Now you might be asking, why do you want 2 illumination objectives? Well, the 2 illumination objectives give us 2 key advantages. It extends our field of view, and it extends our light penetration. So with objective 1, we have a field of view, which is the collimated portion of this light sheet beam that covers about half the sample here in this cartoon example. The other thing that we see is that as the light travels through the sample, it gets scattered and absorbed, and so our illumination on the back side over here is not nearly as strong as the illumination from the front side. So what we can do is bring in a light sheet equal and opposite from objective 2, and now we get around that scattering and absorption by sending a second light sheet with the field of view offset and coming in from the opposite side. We can sync those up together, and now we have an extended field of view combined of both the light sheets and double the light penetration because we're illuminating from 2 sides. So with that in mind, we've got our 2 illumination objectives here. Again, 2 cameras for 2-color simultaneous imaging. The sample chamber, and we've got that big 75 millimeter sample chamber that will drop into the incubation chamber here and physically move back and forth in order to go between the different samples and to create the image. We have a special slide on the sample chamber. So again, 75 millimeters long, a lot of real estate there. What that allows you to do is put tens to hundreds of samples in here. So for example, you can fit at least a dozen 10-day-old zebrafish embryos in there. Or you can get hundreds of organoids in there, either or. But nice thing is you'll just line this with FEP. You have a singular imaging condition here, but you get a lot of numbers. And if you need different imaging conditions for your different samples, we can use the imaging chambers that we showed in the Lattice and actually take up to 3 of those dishes. So you get 6 different chambers, which means 6 different imaging conditions, and can replicate experiments and do screens that way, giving you a lot more control and a lot smaller volume for if you're doing drug treatments. Give you an idea of some of the data that's coming off of it. Here we've got some spheroids that were acquired on the TrueLive, and the relative position of these just shows kind of what well they were acquired in, so as we were going through a multiplexed image. And in the single well, we're able to put a couple Madakas in there and look at their tail extension imaged at 15-minute intervals. So that's it on the TrueLive to start, or to finish. Now let's move on to the QuVI SPIM in upright light sheet for live cell imaging. So a little bit different than the TrueLive in the Lattice in the fact that A, we now have two objectives coming from the top, and B, these are going to be matched objectives. We've got two Nikon 40X 0.8 numerical aperture water immersion objectives again, so it is water immersion. What's nice about having these dual views is now we can have dual alternating excitation and detection from each one of these objectives, which will allow us to create an isotropic sample with isotropic resolution without rotation. The two cameras are coming off the back of here. Hard to see in this particular picture, but they are there. Could be two color simultaneous or one per imaging arm, so one for the left camera, one for the right camera. An upright geometry with easy open sample access, so we can use cover slip mounted samples, SBS plates, 10 centimeter dishes. There's a lot of different options on this. And again, fully environmentally controlled, 6 laser lines in a benchtop contained imaging system. So the concept behind the imaging for the QuVI is a little bit harder, so I wanted to take a slide and go through that. Here we've got our two Nikon objectives that are co-focused in over the sample. And the way this imaging concept works is we'll bring laser in from the left objective, create our sheet of light, and then detect that through our right objective. We call that view 1. Then we'll flip the laser, have it come in from the right objective, have that light sheet travel through the sample, and collect that off of the left objective in view 2. Since these are orthogonal views to each other, what we can do is we've got these overlapping regions, and we can use the information from each one of these regions, computationally plug it into a computer, computationally fuse it together, which is what I was trying to say, and create a single high-resolution isotropic image as highlighted down here. And with the speed of light sheet imaging, we can do this over and over and over again. So here we're looking at a C. elegans embryo that is developing, and we've got our cell bodies marked in red and our membrane marker marked in green. And as the time lapse proceeds, what you'll see is these membrane markers start to brighten up and start to separate as the embryo develops. To give you an idea of the very high resolution of the system, here we have a 3D culture of human primary cells, and we're zooming in to an extremely large image that we were able to slide scan and stitch together. And here we're just highlighting the very high resolution of the system and the ability to kind of see details within the cell. And last but not least, some live cell imaging in a zebrafish brain. So here we're looking at microglia and tracking these microglia over time. So again, to highlight our light sheet portfolio today, so far I've told you about the Invi Lattice Pro and the TrueLive and the QuVI. However, if you had any questions about our LCS or our multi-view SPIM, you're more than free to come find any of our representatives and ask. But the big take-home messages I wanted to send you guys home with is that light sheet fluorescence microscopy is suitable for your everyday imaging. We've got various sample mounts which can be tailored around your different experimental needs, and we have a variety of applications for imaging live specimens with fast volumetric acquisition and minimized photo bleaching. And although I haven't mentioned anything with it, all of our light sheets are compatible with FRAP, photoconversion, ablation, and optogenetics. So if that's something that you could use to kind of advance your samples, those are things that are offered through Luxendo. And I'd like to thank you for your attention. My contact information is there, and I look forward to answering your questions. Thank you.